This is a special episode of the Stem Cell Podcast on COVID-19 research updates with Dr. Josef Penninger. Hey everybody, we are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast for this special episode. We usually culture knowledge and stem cell research, talking to some of the brightest minds in the field, but today we're going a little bit farther afield, talking to a stem cell expert, but about this COVID-19 crisis. Before we get to that, do you love the Stem Cell Podcast and have thoughts on how we could make it even better? We want to hear from you. From now until May 12th, take our online feedback survey and tell us what you, the listeners, think of us. Participants will be entered into a draw to win a Stem Cell Podcast branded Goldie wireless speaker. To participate, visit stemcellpodcast.com/survey. It was about a year ago now that we had Dr. Josef Penninger on the podcast to talk about his research developing human blood vessel organoids. Today, he's back to talk about his work into the SARS-CoV-2 virus. We've also got our usual roundup of recent highlights, this time, though, with the emphasis on the COVID crisis. But before we get to that...
But first, researchers around the world are working tirelessly to study SARS-CoV-2, the coronavirus responsible for the COVID-19 pandemic. And of course, this also includes Dr. Penninger, today's guest. As scientists helping scientists, STEMCELL would like to help by offering relevant products, resources, and support. Learn more at www.stemcell.com/COVID-19-tools. All
right, so as I said early in the intro, we're having an emphasis on COVID-19 research for this roundup. And I'm going to start with just a couple brief little tidbits. It may even be old news by the time you guys hear this because things are moving so fast. But first, I want to talk about what's been in the news and in politics even a lot, this idea of treating patients with hydroxychloroquine and azithromycin. Okay, a lot of people have been proponents of this therapy. Some have said that maybe it's premature or even reckless. And a lot of the reason people think it may be dangerous for some patients is because it's been documented in the past that this treatment can have an effect on the QT interval, right? But let's get back to COVID. You know, there was an idea that was suggested relatively early on, maybe prematurely, the combination of hydroxychloroquine and azithromycin, the so-called Z-Pak, may have a favorable effect on the clinical outcomes, reducing viral loads in infected patients. And because this, caught fire in the news, there was massive, widespread adoption of the regimen just because we didn't really have anything else, right? But both these medications independently, as I said, have been shown to increase the QT interval in a lot of patients, which can lead to this drug-induced ventricular tachycardia or even drug-induced sudden cardiac death, right? So at NYU, a group of physicians led by Lior Jankelson at the New York University Langone Health Center, they, were admitting a lot of patients clearly. And so they sent this correspondence to Nature Medicine to illuminate us on their results. And they were giving a lot of patients this combo, the hydroxy Z-Pak combo, and they observed, not surprisingly, that there was a prolongation of the corrected QT interval. And in a subset of patients, even 10% of them, the QT interval was severely prolonged to a dangerous level and put them at high risk of malignant arrhythmia and sudden cardiac death. Bear in mind, none of the patients died of a cardiac event, although some did die of the disease. A lot of them still remain admitted. But another little insight here that I think was what the takeaway is that in the past, people have pointed towards studies where this combination resulted in a mild prolongation of the QT interval, but this was using young, healthy volunteers. And in this patient population with a lot of comorbidities and the older patients, and especially the ones affected by the disease, they've noted that there is this significantly prolonged QT interval. And so, looking at that discrepancy, I think it maybe reconciles a lot of the disparate results. And I think it really shines a light on how we have to be cautious moving forward with this as a potential therapy. I don't know what you think about that, Arun, but a lot of people have politicized this thing. And I think it's high time that we've had a scientific perspective that really objectively look at the results, and that's here now.
Yeah, man, this is serious stuff. I mean, I want to preface this by saying, like, neither one of us are, clinicians, right? But we do know that there are a lot of drugs out there that can prolong the QT interval, and perhaps there's some association here as well. This is serious stuff. Anytime you're developing a new drug or testing a new drug, this is something you have to evaluate. And in fact, this is something where a stem cell model could help when it comes to evaluating QT interval, evaluating arrhythmias and stuff like that. You want to be able to identify potentially toxic drugs in a safe preclinical setting. So say if you use iPS cardiomyocytes. And in fact, I know people are drug companies and the FDA are actually starting to use those cells to test out to see if the QT interval can be prolonged, or something similar can be prolonged in those cells when they're subjected to drugs like these. So, it's important. It's really serious. I think this is something that's been known with this combination of drugs for a while now. But anytime you are trying to mass use a drug like this, it's something that you have to take into consideration.
Yes, particularly in this vulnerable population, right? I mean, I know we're desperate, but we have to be sensible about this thing. Now, on a brighter note, this is definitely going to be in the news and probably old news. And there's a whole field of, a bunch of vaccines that have been developed, different approaches, RNA-based, DNA-based. One of the early contenders, a more traditional vaccine that was in consideration from a Chinese group, Sinovac Biotech. They've actually just now shown for the first time that this vaccine, which is a more traditional vaccine that's made by chemically inactivating the virus, that they injected it into rhesus macaques and showed that it led to some protection. After 3 weeks after introducing the vaccine, they introduced the virus into the monkey's lungs, and none of them developed full-blown infection. They actually gavage, put the thing in their lungs through the trachea, and they didn't get the infection. So, I mean, that's very encouraging.
Yeah, no, I mean, it kind of points to the importance of animal models in this entire setting. I know there are some animal models out there that obviously aren't as susceptible to the virus in general. So you really have to kind of pick and choose which animals are going to be used for your testing. But this seems, pretty promising.
Yeah, and I think it's as you just alluded to, it's really great news moving forward that the macaque may be a good model. So we can roll out a lot of the vaccines, and this is a nice proof of principle in terms of which animal model is going to be receptive. So that's more encouraging news for all the other vaccine groups there that are working towards a solution.
Yeah, it's not always the easiest thing to house macaques, but certainly if it's being shown that they're useful for these purposes, then perhaps this is a shift we have to make. So next thing is it's a diagnostics paper. It's a CRISPR-Cas12 based detection of SARS-CoV-2. This is in Nature Biotechnology and coming from Boston. Some folks at this is coming from Mammoth Biosciences in San Francisco. Last author is Charles Chu. And we know that testing is really important for COVID-19. You have to be able to identify patients who are positive for the infection so that, one, you might be able to isolate them and distance them. But two, if you have the antibodies, and this isn't a story about antibodies, this is a story about viral detection, but if you have the antibodies, then you want to perhaps use that as a way to perhaps return to the workforce, right? That's kind of the hope. So mass testing is really important, not just for the virus, but also for the antibodies. And these folks are showing that you can use a special version of CRISPR-Cas12, this is a variant of CRISPR-Cas9, to detect the virus really quickly. And they're able to kind of establish a diagnostic test where within 45 minutes, you can use CRISPR to identify the virus in samples. And their positive rates, false positive, false negative rates are really good. Currently, of course, the gold standard is using a qPCR, RT-PCR, and that can take a few hours, up to a day to conduct. But these guys are saying that you can do similar sort of testing, similar identification of the virus using CRISPR in just about an hour. The whole thing is you can modify the Cas nuclease to target whatever portion of the genome you want, right? So in this case, they modified it to identify the SARS-CoV-2 virus, as you might expect. And then there was basically like a system whereby a fluorescent molecule was activated if the virus was detected. So this is important. It's telling us another application of CRISPR. I think this is somewhat similar to what some of the folks at the Broad Institute were doing with their whole Sherlock-based approach, if I remember correctly, where you're using CRISPR to identify viruses, right? But it's got a lot of translational potential. We need a lot of testing. We need way more testing if we're hoping to get around this.
Yeah, it's a great time to be alive, right? In the midst of a pandemic. I know that sounds like I'm being facetious, but it's actually true. 100 years ago, there's 100 million people dead, right, at this point. But nowadays we have the medical wherewithal to mitigate the cost, not saying that this is the Spanish flu in terms of its virulence or the target population that's succumbing, but just saying that we have so many more tools. And we talk about the medical tools in terms of treatment, vaccines and whatnot, but maybe underserved are the technical aspects. There's all these creative, savvy engineering approaches and technical approaches to diagnosing this disease. And like you said, I remember when that initial technology came out from the Broad, and I remember being so impressed by so many aspects of it. It was a simple biological process that had been leveraged to give you this diagnostic indicator with really small input, and it was like off the shelf or something. It was so easy. The idea was that you could disseminate it en masse, and it was modular. So yeah, I'm really impressed by all the savvy the creators out here. People, we talk about creation, you don't really think about it in science. Science has kind of fallen out of that kind of imagination, but this really illustrates the imagination of science and how you can exploit these basic biological principles, tweak them to give you important indicators of, in this case, whether or not people are sick.
Yeah, it's a little bit dark to say, but sometimes, chaos can breed innovation, right?
Yes. And this is certainly a chaotic time. Let's hope we come out of it with a lot of innovation and work forward. I'm going to talk about what this diagnostic test is actually looking at. Again, we were so worried about the sequelae of the disease that there's a lot of research to be done just to understand what this thing is. And part of that, there's been structures solved and the virus and complex with the receptor, but there hasn't been really deep understanding of the actual genomic structure, the structure of this thing. It's an RNA-based virus, so there's that. But it's not, it isn't identical to a lot of other coronaviruses, obviously, in its effect. And although a lot of coronavirus have been studied, and this one is similar in that it's like enveloped. It's a enveloped virus with a positive sense single-stranded RNA genome. It has a relatively large genome, 30 kb. That's relatively large relative to other coronaviruses like SARS or MERS. It has 80% and 50% homology to those, 80% to SARS, 50% to MERS. But as I said, this and other coronaviruses, they have these really relatively large genomes amongst all the other virus families. And like other viruses on cell entry, the RNA is translated to produce these non-structural proteins. And those are the business end of this story. What are those non-structural proteins? They're encoded from all these cryptic open reading frames and overlapping frame shift. It's how you get all that like efficacy out of a relatively small genome of the virus, is you have this, multiplicity of using the same sequence. But of course, also the viral genome is also used as template for replication to get into the cell and magnify itself. And that replication transcription mechanism, although it's been well studied in other coronavirus, it's not clear whether the general mechanism from those other coronaviruses applies to the SARS-CoV-2. So how did they unravel the disease, figuratively and literally? They did it in a unique way, and this gave them a kind of dual insight. They did the classic sequencing by synthesis, or also known as SBS. It's known, you do these little reads of fragments, right? You get a lot of accuracy and you do massive coverage, and then you overlap all those fragments of relatively short read length, 200 to 400 nucleotide read length. And there you get a glimpse of the whole genome, right? And it's very reliable, very high fidelity. They also use this other approach, okay? This is these nanopores. And the nanopores have the advantage, although they're a little bit less faithful, maybe a little more error prone in terms of fidelity, they can give you these really long reads. Also, with the nanopore, you don't have to make a cDNA library, right? Like you do with all the sequencing by synthesis. So you can directly input the RNA. That allows you to get an idea of RNA modifications, because these nanopores now are so sophisticated that they can actually read the modifications. So they had this dual method of looking at the structure of the SARS-2 genome, and they found that, this is very basic stuff, and maybe your eyes are watering a little bit, but it's very important. In addition to showing that there's like these classic genomic and also these 9 subgenomic RNAs, that uniquely the SARS-CoV-2, it makes transcripts of unknown ORFs using fusion, deletion, or frame shift. So these are open reading frames that haven't been diagnosed in other coronavirus. And there's also at least 41 RNA modification sites on the viral transcripts, the most frequent one being this AAGAA motif. Okay, so yeah, again, maybe a little eye-watering, but this is like virus that's doing unknown. It's having unknown effects, right? It's new to humans, and it's killing a lot of us. And the first, I think, step, well, maybe not the first step. The first step would be great to just get a vaccine, but one of the important steps, right, is just to understand what the life cycle of this thing is, understand what the components, what the moving parts are, because clearly that's got to be what's underlying the pathogenicity of this thing.
Yeah, any mechanistic studies that we can do to further understand just how the virus affects the cells are, they're definitely needed, right? One thing I actually was wondering about, maybe this is something you can help answer. When we're doing these mechanistic studies, like, do we have to take into consideration that there are a lot of different versions of this virus floating around? Or are we just assuming that they're close enough genomically that we can pick and choose whichever one we want for our studies? I don't know.
Yeah, I'm not a viral epidemiologist or anything like that, so I don't know. I'm speaking out of turn, but I have seen that in the news. A lot of people are worried about that, at least. Everyone's, I think, in the back of their mind thinking, all the effort we put towards this, isn't it obviously just going to mutate? But my understanding of that is so superficial, but my gut tells me that, immunity is more complex than that, as well as, understanding the structure of this will give you a lot of information of the minor variants in the same way that your immune system can maybe tackle a different strain, although maybe not as effectively. So I do see that there's a lot of concern around that, but every little brick we can put into the wall to keep this thing out of our lives, I think, is going to be really important.
I think everything definitely helps. And I think there is, a significant portion of the virus that is conserved from strain to strain. So, every single thing you can do helps, even if it's not the exact version of the virus that you might be infected by, right? So next, we're going to move on to a couple of single cell papers back to back. I know you have one too that's pretty similar to the one I'm about to talk about. This one's titled SARS-CoV-2 receptor ACE2. We know ACE2 is the receptor for the virus, is an interferon-stimulated gene in human airway epithelial cells and is detected in specific cell subsets across tissues. This is a really huge number of authors on this paper. It's coming from the HCA Lung Biological Network and coming from MIT. I believe the last author of the correspondence is Alex Shalek at MIT. And so this is kind of answering the question of what cells are likely to be targeted by the virus. Of course, we know that the lung is one of the initial sites of targeting for the virus because you're having lung problems, right? But also, it turns out that there are cells in the nasal passages, intestine, that are also more susceptible to infection. And the other thing that they found was that the expression of the ACE2 gene appears to be correlated with the activation of genes that are actually known to be turned on by interferon, which is, of course, a protein that's important for an immune response and a response to viral infection. So they actually explored this a little bit further and did some experiments where they treated cells, the cells that line the airway with interferon, and they found that the treatment actually turned on the ACE2 gene. So it's kind of like a cyclical thing, right? It's telling you more about what cells are being infected. And, I'm going to talk about this in the last story. ACE2 is pretty broadly expressed, across the body. So it's found in the heart, it's found in other portions of the body, but the way and correct me if I'm wrong, the way I like to think about it is, of course, your nasal passages and your lungs are the first things that are subjected to this virus. You breathe in the virus and it's there, right? The virus is in your lungs, and perhaps other cells are being infected too, but the viral load is most immediately present in your lung tissues. So anything you can do to figure out what cells are actually being targeted by this virus, I think is a good thing.
Yeah, I think this is, and I'm just going to jump right into mine, because I think that we're in a phase now where there's a lot of, and I'm not paying short shrift to these investigators, to their work, but it seems like there's a lot of bookkeeping. There's a lot of things that we think, there's a lot of things that we surmise, but we don't know them. And until we know them, there's going to be a lot of people talking a bunch of nonsense at press briefings, okay? And they're going to be suggesting things that, while they may be sensible from an intuitive standpoint, we don't have the evidence yet. So there's a lot of bookkeeping now where we're just showing what we think we know. And this story that I'm about to say is from the same group, the Human Cell Atlas Lung Biological Network. And yeah, like you said, the nasal passages, that's where it comes. If you look at symptomatic patients, you do nasal swabs, you get a higher viral load than in the throat, right? And you see the same thing even in the asymptomatic patients. So like this is one thing. The first point of entry is the nasal epithelium, and then of course later on you get the throat and downstream the lung. So we know, like you said, that SARS is employing ACE2 as a receptor. There's some other receptors that are maybe also involved, like TMPRSS2 has been shown. The protease activity from that is shown to be necessary. So yeah, these authors, the same. It's a huge author list, and they're distributed around Europe in this case, mostly. But it's the Human Cell Atlas Lung Biological Network, just like you. And they're looking to effectively confirm what people have noted by immunohistochemistry. They've shown that ACE2 and TMPRSS2 are expressed in alveolar epithelial cells, in the upper airway ACE2, but there's still some controversy for whatever reason. There's some data sets, that have suggested that these things aren't expressed in the lung. And I don't know, we can maybe talk with Dr. Penninger about some of the controversy about where ACE2 is actually. But, this is kind of putting it to bed, nail in the coffin. They did a bunch of single cell seq from healthy donors. I think this is the key difference in this story, is this is from healthy donors. They were trying to get a kind of baseline, and they showed, ACE2 is everywhere. It's in a lot of tissues. TMPRS is an even broader distribution, so it suggests that ACE2 maybe is a gatekeeper there. But importantly, the nasal epithelial cells, and specifically these two clusters of goblet cells and the cluster of ciliated cells, this is all single cell seq they did. They show that they had the highest expression amongst all the cells in the respiratory tree. So this again suggests that the nasal entry is really, that's it. If you could stop the virus at one place, it might be the nasal epithelium. It seems like that's where the virus gets the leverage. But again, I'm going to circle back and say that this was a kind of baseline study. I think there was kind of double dip here from the HCA Lung Biological Network, where they showed here in healthy donors. And I think in your story, maybe they went a little bit deeper, doing some more experimental stuff to see maybe what was going on in the context of disease. So together, these are really robust and I think undeniable proof that ACE2 is not proof, but very strong evidence that ACE2 is the thing. All right. And ACE2 specifically in the nasal and nasal epithelium, also the trachea, it's the point of entry in all likelihood.
Hey, we're talking with Mr. ACE2 himself today, Dr. Josef Penninger. So I can't wait to, pick his brain about ACE2 and how it's important in the virus. But we know, like you mentioned, it seems like this is really the receptor that's doing the damage here, right? So we've got folks developing antisense oligos against ACE2, different antibodies against ACE2. So that is the focal point here for sure.
Yes, for sure. And the focal point of this interview that we're about to have with Joe Penny is about how he got his thing out in this accelerated review, this preprint about ACE2 and using organoids. And I think, we have this special benefit of having a co-host on this show who pulls his weight. So we're doing a little kind of mini interview here. I think this is an opportunity for Arun to share a story that he's the lead on coming out of the STEMCELL lab. But it's not just the science here that I think is interesting, but it's a little insight that maybe Dr. Penninger can reinforce about how science review is happening, how people are doing work. How do you respond to something like this in the acute phase when everybody's looking at it? Arun, tell us about your story you just posted on bioRxiv.
Yeah, for sure. So we were just talking right now about cells that are possibly being infected by this virus, right? And how ACE2 is really important for the infection. I'm going to preface all this by saying that, yeah, of course we know that there's infection in the nasal passages and the lung. That's kind of where these patients are. These patients are experiencing lung problems, right? But there's an increasing number of people who are also experiencing some cardiac issues too. So arrhythmias, viral myocarditis, or viral inflammation of the heart, these things that are popping up for people who have not only respiratory problems, but also cardiac problems. And so the thought is, if the heart is being infected, is it a primary or a secondary effect, right? Primary meaning the cells of the heart are actually directly being targeted by the virus. And through like some of these single cell studies where we're seeing that the cardiomyocytes, the pericytes, the endothelial cells, different structural components in the heart do express ACE2. But of course, it's tough to do, cardiac biopsies, right? It's really tough to obtain a heart sample from somebody who's alive, because that's somewhat of an invasive procedure. But ideally, you would also want it from postmortem samples too. So we don't know as much about how the cardiomyocytes are being directly infected by this virus, right? And so that's a question that we want to answer. And if you listen to the podcast, you know that my favorite cell type is the iPS-derived cardiomyocyte, right? So it just so happened that, one, I had some leftover iPS cardiomyocytes sitting around in the incubator from a couple different cell lines. And two, we happen to have a very good collaborator nearby here at UCLA, Dr. Arumugaswamy. He's a virologist. He actually had access to the SARS-CoV-2 virus. So we put two and two together, and we decided to basically infect our iPS cardiomyocytes with the SARS-CoV-2 virus as a model to see if human cardiomyocytes might be susceptible to infection. And so we uploaded this paper a couple of days ago. We still have some work to do, and we're in the process of kind of wrapping things up right now. But right now, we're kind of able to show that, at least in this model system, human iPSC cardiomyocytes are susceptible to infection by the virus. We were able to confirm using immunofluorescence the viral particles are actually localizing to the perinuclear regions of the cells. It looks like they are accumulating at the ribosome, so they're being actively produced. So it does seem like, at least in these pseudocardiomyocytes, we harp on this point, right? These aren't perfect replicates of the human adult cardiomyocytes. They are a model, but at least in these iPS cardiomyocytes, which are somewhat immature, they are susceptible to infection by the virus. And they are expressing ACE2 at a low level, but that's probably why they are susceptible to infection. So we've got some more work in the wings. We're doing some transcriptomic analysis as well, but hopefully it's useful data. And that's part of the reason why we wanted to upload this to bioRxiv, right? We wanted to get this out there. Hopefully this is useful information for the community to use. And, I think this is something that a lot of people in the COVID-19 field are doing. They're uploading their biology to preprint servers like bioRxiv as quickly as possible. And for me personally, I think that's the way to go. You need as much data, as much information as you can to help, fight this virus.
Yeah, this goes to congratulations, by the way. It's great work.
Thanks, man.
I really look up to you. I wish I were young again because it's clear that this is a time to be doing great stuff in stem cell science. But, this goes back to the first story I talked about with the QT interval, the observation that you see in patients. It's directly related, yeah? So, this is although you allude to these cells not being the perfect model, it's something. It's something tangible, whereas we can just observe in patients and throw stuff at them and see what happens. It's never going to be a controlled experiment. So you're giving us that insight. The question I have for you, though, is that you posted this in the archive. I'm sure you're going to send it out for review. But in these days where there's so much to learn, there's so much knowledge to be gained, and we know so little. So anything, we keep saying it, anything you could do, any brick in the wall, get it out. But with a story like this, there's so much you could do. You could have 12 figures, if you really were to take this to something close to completion. What's enough? Do you do the seq? Do you have to take it to different patients and infect with the virus and give a potential treatment? What's your goalpost with this project in terms of what do you think is a sufficient threshold that this should, be sent for review, not just posted to share, but be critically reviewed and published?
I don't think there's a right answer to that. I really don't. I think it depends on your perspective. For us personally, we just thought this was useful data and we just wanted to get it out there. And just there's a speed at which this field is moving, right? We have to kind of establish this and send it out for review as quickly as possible. I know I've got another friend who's actually kind of in a similar situation. He's doing some work that's not related to SARS-CoV-2, but his thought is should I post this to bioRxiv? Is this ready for posting yet? Because one thought is what if you get scooped or something by posting something on bioRxiv? I think there was some sort of analysis that showed that the percentage chance of you actually getting scooped by posting on a preprint server is somewhat low. Okay. Look, I understand that there are other people who are doing the same thing. This is a really hot field. I think as much data as you can get, the better, right? If those other folks are posting something on bioRxiv, or if something is published in an actual peer-reviewed journal, tomorrow, I'm all for that. I think this is something that you need for progressing the data in a time like this. Everything helps.
Yeah, there's a glut of data, and it's out there. It's trickling out, at least, these early stories. I mean, this might all be old news by the time you hear it, but maybe we'll come out with another COVID roundup to keep you current. Probably not necessary, but this has been, I think, important for us to just, sink our teeth into what the COVID literature and news is for us. Okay, so maybe it's a selfish endeavor, but we hope you guys enjoy it, and you're definitely going to enjoy what's coming up. Our interview with Dr. Penninger. The kids call him Joe Penny. Maybe not the kids. My kids are going to start calling him Joe Penny. But first, before we get to that, I have a message from STEMCELL. Learn about the features that make air-liquid interface cultures a physiologically relevant model system for in vitro infection studies and how researchers are using them to study a variety of viruses, including the novel coronavirus at www.stemcell.com/COVID-and-ALI. All right, guys, it's my special pleasure for this in-between episode to welcome back to the show a friend of the podcast, Dr. Josef Penninger from University of British Columbia. He's the Director of Life Science Institute at the University of British Columbia, also the Canada 150 Chair in Functional Genetics, founding director at the Institute for Molecular Biotechnology of the Austrian Academy of Sciences. That's in Vienna, Austria, where Dr. Penninger happens to be stuck right now because of the quarantine. They wouldn't let him back into Canada, this poor guy. Anyway, Dr. Penninger's lab uses a variety of tools to uncover the fundamental mechanisms involved in human disease. He's been studying the protein ACE2 for nearly 2 decades, having linked the protein as being the key receptor for SARS and now for the SARS-CoV-2 virus. That's COVID-19. His team has identified a trial drug that can significantly block early stages of COVID-19 in human blood vessel and kidney organoids. Dr. Penninger, thanks so much for being on the show today.
Yeah, great to have me back on your show. And yeah, I got stuck in Vienna because I'm not a Canadian citizen and all the borders came down. So I actually was not allowed to get back to my workplace.
Well, you're stuck there, but I'll tell you, it's a blessing for us because you got some time on your hands. I just have to say, there's a talk I give about this podcast to young people in science. And there's 10 slides, 10 talks, 10 conversations that I've had with scientists. And I introduce you, the title of the slide that you're on is Scientists Are Cool. And that's something I've learned by talking to you. So it's really a delight. You can really talk to the audience. We got a young audience here, also an old audience. And you're a cool guy who's got some really cool science. So I'm not going to waste any more time introducing you. Arun, kick it off, will you?
All right, let's do it. So let's dive right into it. Dr. Penninger, we have to talk to you about your recent study that was published as a preprint in Cell that actually shows that a soluble human ACE2 can inhibit coronavirus infection in engineered human tissues. And it seems like this was a pretty collaborative effort between folks at different institutions worldwide, like Karolinska, STEMCELL Technologies, and UBC. So feel free to talk a little bit about this work. So how were you able to coordinate such a worldwide effort so quickly after the outbreak of the virus? And what do you envision as the next step for a potential COVID-19 treatment using this approach?
Yeah, I'd been actually working on ACE2 for a long, long time. So we co-discovered it in the early 2000s. And I also had been working on SARS. So when the sequence came out for the virus causing COVID-19, it became very, very clear that ACE2 must be the receptor also for the new virus. And at this time, funny enough, I was at the meeting in Barcelona, early February, a meeting on stem cells and tissue engineering and the biophysics of tissue engineering. And it was becoming quite clear ACE2 is the receptor. And I'd been working with these guys in Stockholm at Karolinska on virus infections. We did with them Zika infection, Ebola infection, Marburg virus infection. So Ali Mirazimi, they're running basically the super high security lab for viruses and bacteria for Northern Europe. So I knew this, and Ali and I wanted to do something. And I was in this meeting in Barcelona, and of course, sitting in front of all these people engineering tissues. And of course, it was the obvious to talk to some people and say, hey, why don't we all get together? Let's engineer some tissues. Let's send them to Stockholm for infections. And so I talked to Nuria Montserrat, who had engineered very special kidney organoids. I talked to STEMCELL Technologies, who helped us engineering some actually gut organoids, which we have not in our paper, but we can also infect them. And of course, blood vessel organoids, which I had developed. So within a few days, actually, I put together a team of tissue engineers, stem cell people, people who actually had the virus. So we had the first Swedish virus isolate, and we could infect them. So this is how it happened. In a meeting, talking to people. So.
Yeah, it all starts in meetings. It's why we go to them. But clearly, this is a long time coming. It seems like you said it was over a decade ago that you started working on this. So you were well poised to capitalize on your skill set. But also, I mean, you've had a lot of experience publishing in really high impact journals, and you're no stranger to kind of pivoting your technical skill set to exploit the newest technologies, because that's what the editors like to see, right? They like to see the new tech, maybe addressing an old problem, maybe addressing a new problem, but the new tech is really what drives the interest, right? So you know what it's like to work on something that's quote unquote hot. But the COVID thing, by comparison, is like molten lava. So this is a new experience for everybody. Can you give us a peek into the review process for this? I mean, you alluded to how quickly you were able to assemble the team, but it seems like that was a long time coming. But even so, with all those things in place, the review process for this was really accelerated. Tell us about that. And are there elements of the process you think of that accelerated process that should be conserved, applied generally to all review? Or do you think this is a real special case?
It's a very special case, but what happened, it's probably the fastest paper I ever published in my life. So we started probably 5 weeks before we submitted the paper to Cell. So, I never had in my life a 5-week work, which resulted in a Cell paper. But of course, we addressed a really important question. Is this just a lung disease, or can the virus infect other tissues directly? When we started it, it was very unclear. Now it became obvious that this is happening. So the COVID-19 is not the lung disease, but affects many, many tissues. So we contacted Cell. They, of course, like everybody else, was very interested in top-notch technology papers addressing COVID-19. And we submitted it fast. And then we actually had a very good peer review quite fast. Took around 10 days, 14 days to address the issues of the referees, and then they put it online very fast. So I think it was a very good process. But I'm also sure it's a unique setup, a unique time. Actually, on the same day when our paper was accepted, the second paper accepted in Cell on something totally different. And on this paper, we worked for 10 years.
Wow.
Congratulations on that, by the way.
Thank you. And probably now, after 10 years, nobody will care, because probably nobody will read it, because everybody reads COVID-19 papers.
Yeah, I'm not too sure about that.
Oh, wow. If only the turnaround could always be that quick, right? So as you've mentioned, you've been working on this for a while now, right? So you mentioned you've been working on ACE2 for a long time, and also the virus as well. And understandably, the COVID-19 has attracted a lot of interest in the community, like we were just talking about, right? And a lot of labs are starting to shift whatever they're doing towards coronavirus research. And part of it has to do with funding, right? There's a lot of COVID-centric funding opportunities. But some people are saying that it's actually not a great idea for labs not focused in virology or, ACE2 to actually shift their focus overnight to exclusively COVID-19 work. And I guess the fear is that non-experts in the field might be working on something that they're not entirely knowledgeable about, right? But certainly there's a lot to learn about the pathophysiology of the virus. And I think, you need that kind of diverse, group of people looking at the virus, right? So what do you think about this mass shift in the non-virology research community to actually doing COVID-19 work?
It's really interesting. In the early 2000s, we've published the first paper on ACE2, the knockout mouse, and we published this in, I think, 2002 in Nature, and then had some follow-up papers. I used to be Mr. ACE2, and then, of course, nobody cared anymore because ACE2 disappeared. It didn't disappear. SARS disappeared. It was a nice discovery. It was very nice. But for 10 years, nobody really cared. And now probably ACE2 has become, within the short time, most likely the most researched protein on the planet. Where essentially everybody jumped on it, from MIT and Harvard and every AI company predicting better binding to companies. Every time I get an advertisement from some supply company, they have ELISAs and antibody against ACE2. It's stunning what happened from nearly zero interest in what we had been doing to a global exercise. Literally everybody jumped on it. Of course, not only ACE2, but other aspects of the disease pathogenesis, other aspects of the virus, what it binds to. But of course, ACE2 has become the centerfold, the virus binding to it. So this was really interesting. And I actually get emails every day, like 20 emails from people who have these ideas how to block it. And they come from, plant engineering from Africa. And, one person wrote to me, there's a particular herb which could treat the disease, but you have to cook it in a particular copper. Wow. This is what they get. It's unbelievable. And every company seems to work on this, and every company has a solution for it, from, small molecules to little peptide to RNAi. I am totally mind-boggled by it. 95% of it is most likely total nonsense, but since such a massive amount of clever people working on this, I'm actually very hopeful that something really interesting will come out. And it's actually nice, exactly as you said, that people from different fields jump in, clever people from different fields, because they might actually bring to the topic something we've never been thinking about. So, yeah, it's like the Wild West. Nothing against the Wild West, but it's like it's like a gold rush. It's like the Yukon gold rush. Everybody's jumping in. It certainly has to do with funding. It also has to do that most institutions have closed, right? Mm-hmm. We had to close our institute in Vancouver. The only people allowed to work are actually people working in COVID-19. So that's also basically all the manpower, all the projects are focusing on one topic. And this might have become, I mean, if you think about this, probably the biggest experiment on the planet in the history of our planet, where basically nearly all institutions work on one singular issue.
Yeah, it's a gold rush. I mean, it's a rush. It's a rush for sure. And I think it totally makes sense. A lot of these scientists, they just want to do, I think all scientists want to do something that will be used to help people and to treat disease. I mean, that's like the dream. That's my personal dream. I think a lot of scientists share that, just to have one thing that's used in patients. So I get it. But I mean, you guys alluded to it. You said it straight out. That one aspect of this is that you have all these really smart people, varied expertise, diverse approaches. And there's got to be a few solutions, right? There's got to be, it's not one silver bullet. There's got to be a few different ways that we can tackle this effectively. But, in order to vet those, in order to figure out what's going to work, we need to, assign massive resources, from the money, the people, the patients, the time, the risk, the urgency generally that we're all feeling. Getting these drugs through trials is going to take a monumental effort. Do you think we have to pick our horse, so to speak? We have to, commit to one or few options and put all our weight behind that? And if so, what do you think are going to be the criteria that ultimately dictate which solutions we get behind?
First we need multiple solutions. We need testing solutions. Maybe the testing solutions come from semiconductor research, from sensor research. People actually talk to me, if we could stick ACE2 on some semiconductors, you cuff on it, there's an electrical signal. If you have tests where you go to a restaurant 10 seconds later, you have the virus or not. I'm sure this is part of the solution. There are solutions of making vaccines. I think there are at least 80 companies making vaccines now. There are at least 200 research groups I'm aware of making vaccines. And of course, there we could really ask the question, what's really useful, what's nonsense? Should we put our money on 5 or 6 approaches instead of 200? Just the idea, there won't be enough people on the planet to test 200 different approaches. And will New York use a different vaccine than San Francisco and the Europeans and the Chinese? To be honest, I would have hoped in this scenario that the WHO would take a lead and, focus the efforts in vaccine development. Because now it's become completely wild and everybody does it and everybody has their own ideas how to do this. Again, how do we test it? How do we put the money in? Just to build factories to make vaccines cost $500 million. So then, of course, the efforts of blocking ACE2, so the virus cannot enter us anymore. The efforts to block how the virus replicates. And the many efforts how to block the inflammation which ensues. So COVID-19 is really not the singular disease. It's various stages of the virus exploding, making the first disease, then the immune system kicking in. So there will not be a single solution. There will not be a single magic bullet. There will be not a single wonder drug, but eventually we will come up with combinations of treatment in early stages, mid stages, severe stages, and so, we have to make and put a lot of efforts onto this virus, because that's what the disease tells us. It's not even pneumonia. Today, actually, a paper came out, which was really interesting because it follows our study we published in Cell that one can infect blood vessel organoids. Today, a paper came out that COVID-19 might be actually a systemic infection of endothelial cells in patients who have died of the disease, where they actually find the virus in the endothelium. So there's lots to learn, and of course, many efforts we have to throw at it.
Definitely lots to learn. So, Dr. Penninger, you're a stem cell biologist at your core, and you've joined us on the podcast previously, right? So you mentioned that most of your lab in Vancouver is closed, and unless you're working on COVID-19 research, you're not really allowed to go into the lab. And I think a lot of stem cell labs and wet labs around the world kind of have that same situation, right? A lot of stem cell scientists are having to work from home, but they still want to have a sense of purpose, even though they can't really do experiments, right? So for stem cell biologists who spend so much time in the cell culture hood doing experiments, working from home can be pretty tough. So in your opinion, what can stem cell biologists working from home do to kind of stay productive, even though they're not able to start new experiments from the lab?
Oh, they can do a lot. Read the literature of the last 5 years, which I love to do, with all our craziness of running around and being busy with whatever we are busy with. It's actually nice to calm down, sometimes slow down, and actually read the literature again. So I think that's useful. I know many actually reached out and helping developing tests, using the technologies that they've developed to have better tests for the virus. For instance, I'm seeing this in Vienna now. They developed this super cool new test they will implement in the institute. So I think there are many ways one can contribute. Of course, with our intellects in forums and talk to each other, bring in ideas and so on.
Yeah, it's an opportunity. I'm trying to tell myself this is an opportunity, although I'm borderline suicidal between my kids and stuck at home and not being able to work. But hey, I got the podcast, right? It's keeping me going. You guys said it though, this is a time, an opportunity, let's say I just said. But it's that forced pause. You could read the literature. But a lot of what we're doing is we're talking about COVID. I mean, let's be honest. Every conversation we have, it's about COVID. And I think a part of that whole cultural conversation is kind of reconciling this new world, understanding what the world looks like post-COVID. And part of that is recalibrating everything, every profession. It's like, okay, I got to rethink how I do that. If I'm like, I work in Hollywood, I'm a makeup person, you got to rethink all that, everything, probably. Not everything, most things. So you got to recalibrate. You got to reframe your rationale. You got to reframe the protocols. You got to reframe everything. And we were talking about it. You're saying how there's 100 different vaccines, something like that coming out. And maybe we need to kind of reframe the way we do science. Maybe the WHO needs to step in and centralize all that data so that we can have a one-to-one comparison of the inputs and say, this vaccine candidate is better than that candidate. Who knows? But we got to rethink a lot of things, right? How do you think this is going to change science in a major and lasting way, if at all? Or do you think things will just revert to the norm once we lift the restrictions?
I actually hope that it will change. We had this ultra competitive, social construct of doing science. Have to publish good journals and to have a career, all this total nonsense. So I really hope we get back to a way of, where collaboration counts, where working together counts, where sharing data actually counts instead of, I'm the first one and this is what counts for our careers. So So I'm actually completely on your side. So I think we have to reevaluate what we're actually doing and how we do it. And my wish would be that we work much more together. We are much more collaborative again. We learn from each other, and not just for science, for many other aspects of life. I like to, on the side, I like to start companies. And last year we started a new company, and we had this super cool technology, how to actually find weak spots of viruses. We can actually find Achilles heels of viruses nobody else can find. So half a year ago, I would go to investors and ask them, if they would give us some money because this is really cool. We could find the next generation of antivirals. They all threw me out of their offices. There's no money to be made. Let's work on the next cancer, because we can make more money. We can calculate how many people get this. There's a market analysis of health. When I listen to these talks, then I could nearly throw up because it's human health. It's people. We are not part of a market analysis of people from the Nasdaq. I mean, seriously. And so, we had actually technologies and nobody financed it because they thought they couldn't make any buck out of it. So I really hope that things like this also change, that our world, shows a little more solidarity, that we actually do things which doesn't make people who are already very rich even more rich, help all of us. So it's my wish, which is coming out of this.
Do you think, because you said you would like to come back to a time where we're working together again? And I have to say, I mean, you're still a young man. You're not that much older than me, but I can only remember it ever being super competitive. But was there a time when you were starting out that you think it was more collaborative? And what changed? Was it the market? Was it the money thing that you're alluding to that kind of tainted the collaborative effort?
I actually, when I started out, I was super competitive. I mean, I still am, otherwise I wouldn't be in my position. It's competitive in a way to move things forward, not competitive in a way that, I can only move forward if I screw somebody else. This is not what I understand under competition. But when I started, I also had this idea. It's my new idea. I cannot tell anybody because if I tell people too early, they might steal what I'm doing. And once I had the pleasure actually to run into Oliver Smith's dinner with Oliver, who won the Nobel Prize for making knockout mice at homologous recombination and bringing it together with stem cells. And we discussed really this topic. Me as a young researcher just starting out and trying to make my way into the big game. And so I asked Oliver how this is, and he said, if you talk to 10 people, 2 people will steal your idea, but 8 people might give you a much better idea. So there's actually no choice. Talk to people, work with them, know that, there might some assholes out there who might take your stuff. But I think most of us are quite reasonable and want to work together. And if you tell me something, and we're all honest and frank about this, I might give you a good idea and good suggestion. And at the end of the day, we advance much better together. So this, I think, was my defining moment where I realized it's okay to be competitive. We all have to be competitive to a certain extent, if it doesn't hurt others. After this, I waited for half a year for other people to catch up with our work, to publish back to back, because I thought sometimes it's more important not to be the first one, but to make sure it's real. If another group comes up with the same solution. So, yeah. So
I guess kind of on that topic of competition versus collaboration, I think something recently that's been happening to facilitate collaboration is people have been posting their manuscripts on preprint servers, right? Like either a CellPress preprint server or bioRxiv. And I think even your manuscript was on a preprint server before publication. Do you think ultimately that's a positive effort in the scientific field? And do you think that's something that's going to carry on?
Honestly, I don't know. Maybe I'm too old-fashioned for this. I'm not totally sold on it, to be honest. But when I talk to young researchers in my institute, they really like this and they really believe that it's the future. So maybe I'm just too old-fashioned and growing too old to understand it. It certainly, and I'm sure it will be the future because many more people are using it, and I'm not totally convinced by it because, of course, there are lots of things which are posted, which are not peer-reviewed.
Yeah.
And I can tell you from COVID-19 research and ACE2 research, something I really understand. There are lots of paper preprints, where people claim ACE2 is actually not expressed in the lung. So we cannot explain any of the disease, having a bad antibody, but because of COVID-19, this, creates a lot of media, and all of a sudden there's this whole, online Twitter exchange. ACE2 cannot be important because somebody in an uncontrolled fashion published some nonsense. And so that's what I mean. We have to always take this with a grain of salt. In particular, and I realize this now with COVID-19 research, the research, and we all know this, depends on good reagents. And we all know there are bad antibodies out there, there are bad reagents out there. But with COVID-19, anything you could, for the last 2 months, you could publish anything. Mm-hmm. If there was ACE2 or SARS virus or, COVID-19 on the title of your paper. So at the end, we still need peer review, I believe, to make sure it has been done properly.
Yeah. Yeah, I think we have to strike that right balance. So shifting gears a little bit, not too long ago, you moved to Vancouver from Austria, right? So you're heading the Life Sciences Institute at UBC, and you've been back in Canada for a little while now, although, I think now you're back in Austria, right? Since because of this whole situation. So you're a world traveler and you're kind of a new resident of British Columbia. So how would you evaluate how the government and the people of Vancouver have responded to the COVID-19 pandemic? And how would you compare that response back home in Austria?
So I might step on some people's toes now here, which is probably why you asked me this question. I actually was here when the whole COVID-19 exploded in northern Italy. And I don't know if you know why it actually happened in northern Italy, because the fashion industry. So the fashion industry has lots of connections to China. In northern Italy, there are around 500,000 Chinese actually working for the fashion industry, making shirts and so on. So in all the global pandemic scenarios for Europe, northern Italy was basically always the first place where it would strike. And of course, there were soccer games, Champions League. There's this famous soccer game of Atalanta Bergamo in the Champions League playing against Valencia, which was played in Milan. And I think half of the Valencia team was infected. So this is how it came from Italy to Spain, how it hit actually Spain and, of course, France through the fashion industry. And then it hit Austria. People going skiing, the Italians coming skiing. In Switzerland, I think there are 200,000 Italians who come over the border every day to work there. So this is basically how it spread. And in the Austrian ski resort, people partying in the evening in close range. Austria basically infected all of Germany and Northern Europe. So, of course, not all of it. These guys going skiing and in the evening, have the big parties on the ski resorts. And I think probably everybody in Iceland was in the same place, in the same bar in Austria who got the disease at the end of the day. So this was quite interesting to see. Now, having seen this, I actually was in Austria, and it was already full-blown in Italy. So the Austrians still, kind of ignored it. This cannot happen to us. So I still remember I went to go to a museum, I saw an exhibition, I went to the restaurant, there were already many deaths in Italy. So I think that's probably our human nature to ignore things. A week later, the Austrian government shut down the entire country, police on the street. Nobody was allowed to go out anymore. It was really, really interesting to see this, from going to a museum to, being controlled by the police. And actually, you can get fined. So if you're too close to somebody else, you could get fined for 500 euros, things like this. But now Austria is the first country, I don't know if you know, which is opening again. So since 2 days, the small shops are open. Actually, today, our institute, the old institute that started and founded, opened again. Next week, the shopping malls open again. So mid of May, the hotels will open again. So everybody of the world is actually looking at us. So at the end, the Austrians reacted late, but then they reacted very fiercely. And at the end, we have 10,000 cases, very few deaths. So we're probably one of the best countries which went through this. Now to Canada. Because I lived through this and, told the Canadians, this is not a harmless virus, take this really serious. And, like the Austrians, they completely ignored it. This cannot happen to us. It's no problem. This was not a good experience, I have to say. And also they knew, for instance, that the way to do this is we have to develop tests, early testing. We have to test lots of people. We had people I know who had typical symptoms, wanted to get tested 3 times, could not get tested, they were sent home. So honestly, what I know, I was not particularly convinced that the response in Canada was at an appropriate level. Of course, in retrospect, we're always more, it's always easy to criticize. But of course, they could have learned from the others. Having said this, we have not learned from the Italians, and we did probably the same mistake. Anyway.
Yeah, it's tough. I mean, I've heard the expression, darned if you do, damned if you don't. I'm hoping we're not damned, but I think there's light. I think we're getting near the end, and I hope the trends continue on a downward trajectory. Just to close out here, I want to ask you what's next. I mean, this was a big story, but you bookended it well there when you said you had a story that was published essentially the same week or day in the same journal, but it was in review for a decade. So, I mean, this is a big story, obviously the preprint, but unlike that decade-long review story, which languishes for so long that you've fleshed it out and you have next steps there. Like you've already moved on a lot of times by the time a paper is published and you're exploiting with the gains from that paper and applying them already in the next step. But here, the paper's out. You just started working on the thing. I mean, you've been working on it forever, but you just pretty much picked up this COVID thing and it's published. What do you do now? I mean, there's no languishing. The decks are clear and you got to move on. You talked about like the vasculitis thing in the blood vessel, how the centrality of the blood vessel endothelium to the disease. Is that where you go, or do you just pivot to something else altogether? You got some on the back burner that you bring back to the front. Tell us what's next.
So the next step. So for instance, we are making now a fully humanized ACE2 knockout mouse. So since we had made the first ACE2 knockout mouse and we have ACE2 flox mice, so we are now getting a mouse from China, which is human ACE2 under the endogenous mouse ACE2 promoter. So we will cross them together to have really a mouse model with human ACE2, because it seems the virus only needs human ACE2 to enter the cell and then it wreaks havoc. And of course, then we can do a lot of experiments. Of course, we'll share these mice with everybody who wants them. Then we can do live infections. We can really study the immune system, therapies, vaccines in an animal model, which we can manipulate, really study, how immune cells, T cells, B cells, innate immune cells might be important, how the disease really develops, what we can do against it. So that's one thing we are doing. Of course, this is not novel. Many others are doing. I know the NIH is doing this, and many others. So, but we will have the possibilities, and of course I will share it with everybody. In Vancouver, we have actually a particular level 3 laboratory where we can do mouse work with the live virus. The same we are doing in Stockholm at the Karolinska Institute. So they have this high security lab, which is of course also very important, because at the end, we want to have a real life virus infection. The other thing which really interests me, which we are doing in our group now, is after we published our organoid paper, I'm aware of at least 100 other groups who do exactly the same now. So, from the Harvard guys. And of course, they have a real power and lots of money. So how do we survive in a competitive field like this where everybody does the same? So what we're actually doing, we are moving on to the next step. We are developing blood vessel organoids under diabetic conditions, which we published last year. And also kidney organoids under diabetic conditions, because diabetics have much worse disease. And so, model in, stem cell derived organoids, diabetes, disease conditions, and then see at the molecular level how this would actually affect why people who have diabetes get worse disease. Maybe the virus binds more. So that's what we're really doing right at the moment, to look at single cells, how they get infected and what happens to the next cell beside it. So to gain some information, which of course many other people are doing at the moment. Where I really spend a lot of time now is to help out in setting up a clinical trial, because the molecule we used in our paper has actually already been tested in 89 humans. This all comes back to my old work in SARS infection and ACE2, and we could explain why the SARS virus became a killer virus, because it binds to ACE2, and ACE2 gets downregulated, and ACE2 is a protective factor for many, many tissues. And that's exactly what we see. ACE2 is in the gut, in the kidney, in the heart, in the lung, in the nose. So we can explain all the distribution. And this virus is a real crappy virus because it hit a molecule and shuts it down, which normally protects us. So based on this, we developed this medicine. Many people got it already, healthy volunteers and phase 2 trials for people with lung disease. And so we're actually setting up now a clinical study, a very carefully set up study, a placebo controlled, double-blinded, so not some fast shot, 10 patients and it works, and the president says this is the next wonder drug. So, because at the end of the day, we need to know if it works or doesn't work. It's totally useless to come up with anecdotes, even if it works, but it doesn't help anybody. So that's what we are doing and have been doing. Talk to people, advise the Austrian government, talk to ministers, funding agencies to actually give us money to do the clinical study, and also help a little bit setting it up. So I've been in contact with intensive care physicians from China, from North America, from Europe. The trial has been now accepted, so maybe even today or tomorrow, the first patient will be treated in Austria, in Germany, and in Denmark.
Wow.
And we might open additional sites probably next week in the UK and in Spain. And so that's how I spend my time. And I have to say, besides, doing basic research, which I love, and this is where I come from, this is a completely different ball game. You're talking about patients who are very sick, exclusion criteria, inclusion criteria, who can be treated, who should not be treated. So I'm actually learning a lot. And sometimes when we have this vision as scientists, this should work the best here and there. There are, of course, the realities of clinics, of clinicians taking care of people who are dying. They cannot, to centrifuge a tube is not their priority. So they have to save lives. And so that's what I've been doing. And from basically 7 a.m. in the morning to 2 a.m. the next day for the last 2 months.
Wow. So, guys, what are you doing during your quarantine? I know Arun is learning the guitar. I'm growing out my afro. But Joe P., he's doing all that, people. So you got to measure up, pick a craft or something. You got to have something to show for it. Dr. Penninger, thank you so much for joining us. I hope we can have you back again and before a year, because, it was a year this month, actually. You were with us last. You've had about 20 papers since then, made a lot of big strides. If they ever let you back home to Canada, I'm sure you're going to knock out this COVID thing next, and we'll have you on when you do that. Thanks again for this chat. It's been a lot of fun.
Yeah, thank you so much for having me on.
All right, you guys, that brings us to the end of our COVID special. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to the interview and roundup papers. You can also reach out to us on Twitter at StemCellPodcast or by email at info@stemcellpodcast.com with feedback or to suggest guests. We'll be back in just a week because this was an in-between episode, so you can hear us then. Until, thanks for listening.